Thank you, Carolina, and thank you for letting me present our platform here at now it works. Yes. Okay, I call this CBMR Metabolomics platform. It's not just measuring, and I hope you will find out why I call it this title a little bit later. But I will start actually to just say some words about CBMR actually. So the Novo Nordisk Center for Basic Metabolic Research is organized around 4 different programs. And I listed them there. It's integrative metabolism, environment influences, the human genomics, and metagenomics in metabolism, nutrient and metabolite sensing. And those are the 3 that are in the tower here at Panum. But we also have the 4th is called metabolic science and culture. That's a bit outside at the medical museum, actually, part of it. If I now get this kind of laser pointer actually to work here. Around these different programs, there are also different projects ongoing, and we have a large number of different group leaders around these programs, of course. Besides the programs, we also have a number of different research facilities at CBMR. And I list them here. You have their own metabolic phenotyping platform, Metabolomics platform, single cell omics platform, and computational chemistry unit. And the newest one is actually the Metabolomics platform, because we started around 1 year ago. We started building up the Metabolomics platform, and I will say some words about that right now. So we have some sort of overall aim that's being flexible and look into the future, and that's of course a strange thing. But what we would like to do is actually standardize in a flexible way, and the reason for this is just that at CBMR there are very many different kind of projects ongoing. So that's not one project that are similar to each other. Therefore, we need to be very flexible. And that's a reason I'm just saying it's not just measuring at our platform. We would like to actually be to say that it's the research question that to determine how we do the method actually, and which methods we are using. We have also, I think, a very close connection to the researchers. We really want to explain for you what can be done, or even more important, what cannot be done actually. So that is an ongoing discussion all the time within the project. Important part is to be part of the whole process, not just being like a normal service unit. We like to be involved from the experimental design to results, if possible. That's not always possible, but hopefully many times. And then to have a continuous discussion on what is needed, because we don't want to be something that are on the same stage the whole time. We actually want to develop. And I think personally that it's the users must be involved in how to develop the platform. If I say more, but what we're doing, of course, we are having metabolomics, lipidomics analysis. And when I'm saying that, I'm saying that we have untargeted methods. For that, we have 3 instruments mainly we're using. We have 2 timsTOF Pro instrument from Rutger. We have also a GCMS from LECO. These are currently our basic instrument for the metabolomics, lipidomics analysis. And what we do there is, of course, we are trying to screen for as many metabolites as possible. For the LC, we are mainly using reverse phase chromatography because we are planning to include helix chromatography also. Some people's favorites, some people's are not so favorite to helix, but we are trying to do that also. So this is the current setup list. We are also, and this is an important part, we are also doing targeted method. I call it target metabolomics, an expression I don't really like, but targeted methods for a large number of different metabolites. For that, we have a triple quad from Waters, and I was saying that we are also using the previous instruments from the GC-TOF and the LCMS, the timsTOF, also for targeted methods. So it's more or less, it's not only the triple card we're using for target methods. And what we do there is, of course, we are trying to focus on specific metabolites, and that can be bile acid, TCA cycles, or amino acids and others. But I would say that what we're actually doing, that we are trying to focus on metabolites the user wants to be quantified. And this is very many different kind of compounds that people here at CBMR wants to be quantified. So we really need to be flexible here. We are not actually defining ourselves always what kind of methods we are developing. Actually, it's quite diverse. Good thing here is, of course, we can use calibration curves, and we can actually do absolute quantification of metabolites there, if needed. Another area that we are trying to develop here is, of course, to determine fluxes of metabolites, because in biology and the need of actually determine not only the static levels of pools, it's also important to follow turnover of metabolites one way or another, flux of the metabolites. And that is, of course, we're using carbon-13 labeled or deuterated or 15-19 labeling compounds for that. This is just an example, of course, when we have treated pancreas cells with the carbon-13 labeled glucose, and you can also follow these traces into the TCA cycle fairly easy. This is an example from a GCMR spectra. This is the library. Might be difficult to see on the screen, but it's the library for carbon-12 citrate. This is the labeled one, actually. And this is the ion traces then. With that, of course, one need to actually have some sort of idea how to actually calculate incorporation of carbon-13, and we are using something I call taking from my former working place. So this is the general workflow of the platform. So what we try to do, we got request, of course, and then an important part here is actually to have a very good discussion with the researcher to define the project. So what is it? What are they actually asking for? Are they asking for screening methods like metabolomics or lipidomics? Are they asking for targeted analysis? If they are mainly interested in some of the metabolites, or it can be amino acids or fatty acids, whatever. Do we need method development to actually be able to help them? And what is it? Is it a part of important to identify novel new compounds or anything like that? Or they are not interested in that? And this is, I think this is the base for actually our workflow here. And the reason I say this, that to define the project together with researcher is that it's not always this case that the screening methods are the important part here, because people say many times that, well, we need to do metabolomics analysis, but actually what they are after mainly is a few compounds in their samples. And of course, then it's much better for us actually to develop a method for that. Sometimes we can, of course, also give them more data than what they are asking for, but I think this definition of the project is the key point here. And then, of course, we take a decision about the project, and then it's about how easy is it to use, and that to run the project depends on the size, the methodology, and timing, of course. But usually we can come up with a strategy for this, which I think most of the time people are happy with. We're putting it in the queue, and then we are running the project, and then we are actually then giving the results to researcher. And then it continues this kind of thing, because what we are actually giving to people, of course, we're doing some statistics, we're trying to put that into some sort of biological context, but this is, of course, a discussion with a researcher and its group. because what we need here is, of course, we are not always the expert on these projects, so this must be some sort of discussions with the project leaders. But we usually think that the project is, of course, not ending there. So what is more needed for us? Do we need more statistics? Do we need more bioinformatics? Do we need new analysis or additional samples? So it's a continuous discussion with the group after the first analysis is done, at least. And with that said, I don't think we don't want to see us as a traditional service platform. Our aim is to be a truly integrated in the research projects, and that is important for us. We are not a CRO company. We are a research platform with close collaborations with the users of the platform. A platform without staff is, of course, of no use, and so this is our current setup. We have 2 staff scientists on the MS side, we have a lab technician, and we have a bioinformatician that will start from 1st of March. I also would like to add that we have also PhD students and postdocs that are really closely associated to the platform. And many of the things we are developing or using and doing at the platform are many thanks to them also. So although these persons here are the key points, key staff at the platform, those who are students and postdocs that are associated to the platform are also very important for the development of the platform. And then since 1 month, we have also a steering committee. We have never had any meeting yet, but we will have it, of course. And that is, well, I'm chairing that. But then we have 3 members from CBMR. That's Jonas Trebak, we have Atul Deshmukh, some of you know, and we have Thomas Kilpeläinen. And then we have an external member also, and that Douglas McCloskey, of course, a member of the CLIME. And I think that's really good that we have an external member of this platform also. The idea with the steering committee is mainly to take decisions on overall decisions, and also be a support for the platform, actually. What is needed, what should be focused on in the future, and things like that. The focus areas, Karen, I say the focus areas here, this is a bit perhaps not correct, but it's what we are working mainly with now is, of course, put a lot of effort into metabolite annotations in metabolomics. This is one thing, is to increase number and metabolite and annotating metabolite, but also trying to actually identify novel compounds, because there are several products here at CBMR that are involved in this kind of identifying novel bioactive compounds. So we need to have a strategy for that. We have targeted methods. This is an important part. Many of the research projects are focused on specific metabolite classes, for example, nucleotides, short-chain fatty acids, bile acids, etc., etc. So this is something we have methods, or we are developing methods. In the future, and this is something ongoing now, is, of course, that we are trying to develop large-scale metabolomics and lipidomics. And the reason for this is we have several codes, I would say, that need to be run at the platform. And for doing that, we need to identify or actually prepare us for automate the workflow from sample preparation to data analysis. And this is something we are now starting up. Yeah. And then, of course, already mentioned, which I think is personally very important, is to continue with the developing of the fluxomics part. So I would just say that we are definitely open for collaborations. I'm trying to actually get contact now with, I'm fairly new here, so I need to have contact with the Copenhagen cluster. There's a lot of metabolomics ongoing in Copenhagen, but of course I'm also involved, open for other types of collaborations with the platform, actually, to share methods, to share strategies, workflows, for example. That is, of course, possible. And I also have still my affiliation to Sweden, actually. So we are also collaborating quite a lot. And this is the end for listening.